Hey, everybody, good morning. My name is John Galvan. I am the Project and Research Coordinator for the REEU program. So what is the REEU program? The REEU program is REEU. It's actually an acronym. It stands for Research and Extension Experience for Undergrads. This is a grant-funded program funded by the USDA in collaboration with the UMass Fruit Tea, Stockbridge School of Agriculture, and the UMass, or University of Massachusetts Amherst. Like I said, I am the Project and Research Coordinator for the department. Along with my leadership, our two directors, co-directors, Dr. Jaime Peñado and Elizabeth Garofalo. They also work with me on the UMass Fruit team. This year, we were very, very lucky. We're actually in our fourth year of this program. It's a 5-year grant. And this year, we actually have alumni with us. Mentors that were hired on this year to help me in my job. Samina Mian and Caitlin Elishowskis.
Hi, all. My name is Caitlin Elishowskis. I am, along with Samina, in the same year. We were 2023 REEU interns. We both had the pleasure of coming back this year to be the mentor and help everyone out with their projects. We also put on a podcast and we put on a magazine, which we do have over there. So you guys should check that out before you leave. We also have a few QR codes lying around. So if you guys wanted to check out our website and then view past projects, future projects, and maybe future opportunities, please do so. Thanks.
And like I said, thank you both. I could not have done this program without you guys. Your support was amazing. Thank you so much. So REEU, what is our mission? What is our goal? What is our intent? Our main goal is to provide research to undergraduate researchers that have never had research experience before, and it's mostly focused in agricultural systems. And on top of that, we allow them to actually participate in their own research, their original research that they thought of themselves. And then we also show them how that research applies in agricultural systems immediately. The research that we do at the extension is stuff that can be applied today. It is not research that comes 2 or 3 years down the line, which is why I like my job so much. And this is the breakdown of what they went over this past summer. It was a 14-week program, broken down week by week, and each day, or two of those days, they would be doing workshops along with me, whether it's career development, writing, team building, scientific writing, all that kind of stuff. They did it with me personally. I gave them lectures, hands-on experience on how to do that. And we also went on field trips every single week. And then they had 1 or 2 days of independent research where they worked on their own personal projects. And I am very proud of these 8 individuals because I have individuals from all different types of backgrounds. Some are sustainable agriculture, some are computer science, some are resource economics. I have 2 microbiome majors that I'm very proud of. These are all the places that we went to go visit. And I want to give a special thanks to Plumbrook Farms, Park Hill Farms, Headwater Cider, Red Apple. Can somebody let him in, please? Stockbridge Floral Design, the Cranberry Station Camp Howe, Norris Farms, APHIS is great, Student Farms, and everybody else that's on here. Thank you so much for your support. We couldn't have done this without you also. Thank you. I also want to stop and thank all these people that is on this list. All right, moving on. These 8 individuals I have worked with over the past 14 weeks, I am very, very proud of them. I've got to know them a lot personally, and they may look like this, but in reality, they look like this. I've got to know them personally, and they are very, very strong, very, very fine individuals that I'm positive are going to do great things in the future. The scientific community also. Moving forward, these 8 individuals are going to present their research for you right now, and our first person coming up is Raphael Amsili. He is a plant and soil science major. He is a senior. He's going to be graduating 2026. Could you please come forward?
Yeah. Hi, everybody. My name is Raphael Amsili. I am really excited to share my summer research. As John said, I'm a plant and soil science major. And personally, I've always been very interested in plant microbe interactions. So I'm excited to share my project on plant fungal interactions called Does Fungal Nitrogen Transfer and Carbon Availability Shape Brachydipodium Growth in Vitro? So to start off with a little background, symbiotic plant microbe interactions are definitely a very hot topic within the plant science and agriculture realm right now. Microbes have been shown to improve growth outcomes, biotic and abiotic stress, as well as nutrient acquisition. This nutrient acquisition was a piece that I was personally very interested in. So potentially in the future, if we can harness these interactions, it could lead to a lower input agricultural systems, which would be a great benefit to farmers and the environment overall. Yet there's a lot of research that needs to be done. Microbial dynamics are very complex. They vary from place to place and are very context dependent as well. And this is important because there are a lot of biological amendments and inocula that are hitting the market, and we want to be sure of their efficacy before people are spending money on them. So, as I said, I was very interested in kind of the nutrient acquisition portion of this, and arbuscular mycorrhizal fungi, or mycorrhizal fungi in general, are very well known for their nutrient benefits that they can give to plants. So you kind of can see these exchange sites within the root cells, in which the fungi extends the plant's reach for inorganic nutrients such as nitrogen and phosphorus in exchange for the plant's carbon. However, there are another really important class of plant-associated microbes called endophytes, which are largely defined by their existence within plant tissue without causing a pathogenic response. So I am studying a root endophyte called Serendipita besseyi, which is in the genus Serendipita, which is in the Basidiomycota phylum in the Sabicinalis order. So it's in the same kind of group as a lot of the common mushrooms one might see in the woods. And this genus has been purported to have and shown to have a lot of benefits to a pretty wide range of crops. So it's of very interest to us. And this is the first North American isolate of this kind of larger species complex, Serendipita vermifera. And this is really exciting for us because the two most well-studied isolates are from Australia and India. So this is exciting for an application to North American agriculture. And yeah, so this has been shown to have some benefits on crop growth and nutrient stress. The preliminary study using winter wheat showed that Serendipita promoted plant growth under both nitrogen and phosphorus limited conditions. However, it remains to see if this kind of operates similar to the mycorrhizae I discussed earlier, in which they're directly transferring the nitrogen to the plants. And so that's kind of my goal for this experiment, is to see if Serendipita besseyi can promote growth outcomes of Brachypodium, which is a model grass species used in research, through nitrogen provision. And also to determine if non-plant carbon by Serendipita alters growth outcomes and fungal colonization. The secondary question, as I mentioned, these interactions are very context dependent. Arbuscular mycorrhizal fungi, as I mentioned earlier, cannot, obtain their own carbon source. They rely on the plant, and they can't exist without the plant. However, Serendipita can digest an environmental carbon source. So I'm interested to see if it will kind of have a preference towards that environmental carbon source over the plant and not want to give up any of its nitrogen that it's obtaining. So I ran a split plate assay, which is essentially a petri dish that is divided into two compartments. And then there's a little agar bridge connecting the two compartments. So this is a really cool assay because it allows us to control for nutrients in the different compartments. And the bridge is only accessible by the fungus. So the fungus is the only thing that has access to the nutrients that are in the opposing compartment. And then the measurements I'll be taking at the end will be using kind of root and shoot biomass as a proxy for nitrogen transfer and looking at colony diameter and percent colonization. Just to kind of run through my treatments. Treatments 1 and 2 are just fungal controls, kind of getting a baseline for what fungal growth looks like in kind of either full strength media or minus carbon media. And by the way, I'm using a MS media, which is a common plant growth media. Treatment D1 is a brachypodium control, which is brachypodium growth in nitrogen limited conditions. Treatments 3 and 4 are also brachypodium controls in full strength MS and then low carbon MS. And then 5, 6, and 7 are my experimental treatments. So this is the treatments where brachypodium is being grown with Serendipita. Treatment 5, there is no access to an environmental carbon source. Treatment 6, Serendipita has direct access to an environmental carbon source. And then treatment 7, Serendipita has delayed access to an environmental carbon source. So this is kind of just an example of like what this looks like in person. You have the brachypodium as well as a 6 millimeter plug of Serendipita inoculated agar, and then the agar bridge. So what's my overall hypothesis? So based on the research that was done with Serendipita besseyi, I am going to hypothesize that grown in association with Serendipita, brachypodium will show greater root and shoot biomass than brachypodium grown in a nitrogen-limited environment in isolation. Secondly, Serendipita will promote greater root and shoot biomass with brachypodium in a carbon-limited environment than in a carbon-rich environment. And both of these kind of, the first is kind of pertaining to nitrogen limitation, the second hypothesis more pertaining to carbon availability. So kind of let's look at some results. So I'll kind of take us through these results in respect to both of my hypotheses. So we have dry shoot biomass on the left and dry root biomass on the right. So in terms of nitrogen limitation, I hypothesize that Brachypodium will have a greater growth outcome when it's associated with Serendipita. And so I have my treatment here, treatment B, which is grown in isolation, and then my experimental treatments where it is inoculated. The letters signify statistical significance. So things of a similar letter, sorry, things of a in the same letter don't show any statistical significantly difference. Things of a different letter do. So we're not really seeing any statistically significant difference when Brachypodium is grown in association with Serendipita. In respect to my carbon limitation hypothesis, I hypothesize that treatment 5 will have the greatest growth in a carbon limited environment. However, there was also no significant differences found there as well. So I also wanted to look at the roots and see what's kind of going on there. So I wanted to quantify root colonization, which is done through a grid-line intersect method. Grid-line intersect basically is a simple method where I'm placing roots on a grid-lined microscope slide and basically assessing at 16 random intersections whether I'm seeing the presence of fungal structures or not. So it's basically just a yes, no, and then we can get a sense of percent colonization by the number of yeses over the total number of intersections and, kind of get a rough percentage. So this would be a great example of like what a yes would look like on the left. You can kind of see the grid line in the background. So on the left, I'm seeing a fungal structure there. And on the left, yeah. And on the right, I'm not seeing any fungal structures. So this was pretty interesting because I hypothesized that low carbonability would actually promote greater presence of fungal structures. And I'm actually seeing that treatment 6, which was having direct access to carbon, as actually being the best, the most colonized treatment. And this was really interesting because also with respect to colony diameter, this was also significantly slower growing than the rest of the treatments. So potentially kind of my idea is that colonization was more so a function of colony density rather than carbon availability. So this was a pretty interesting result that I got. However, there are definitely some sources of error, some limitations that I kind of want to troubleshoot and change in the future. The bridges are very finicky to work with. They kind of would get messed up and dislodged, and I would have to, put them back. And then also kind of a relatively short amount of time for growth. So this experiment was kind of seeing, with fungal access to the opposing compartment, would growth outcomes improve? However, in both my carbon limited treatments, both did not reach the other side until the last 2 weeks of the experiment. So potentially there wasn't enough time for the fungi to provide the nutrients to the plant. Seed-based contamination most likely was due to a sterilization protocol. So we did have a little bit of contamination. However, we were able to troubleshoot this, which was great for future experiments. And we showed that a secondary bleach treatment after vernalization was actually very effective. Then the limitations of grid-line intersect as well is I'm just assessing whether or not there are fungal structures present. I think it would be great in the future to grade what kind of fungal structures I'm seeing rather than just a yes or no. So why does this matter in the larger context of agriculture and extension? So this is actually a Serendipita inoculum that is coming on the market from Premier Tech. This is kind of one of the many examples of microbial inocula that are on the market right now, but this one is Serendipita specific. And we're kind of not really sure of their efficacy yet. This research shows that there, is a lot of conflicting results sometimes, and I don't want growers to waste money on products that might really not work. The science is really not there yet, I would say, and most likely these simple suggestions of, improving soil organic matter to maintain healthy soil ecology would be the best suggestions that I could give. And then why does this matter in the context of fruit production? So serendipita is known for evolving ericoid mycorrhizal interactions, which are interactions with ericaceous crops, such as blueberries and cranberries. So this potentially in the future could be a tool to improve blueberry and cranberry growth. However, it could also just be unnecessary. There very well could be native serendipitous species that could be isolated in these environments. So they could be already doing their thing and could be interesting to kind of see what we could find there and what these species are doing. So potentially all very interesting things for the future. And yeah, that's pretty much it. I really wanted to give a huge shout out to the Hestron Lab, both Dan Myers and Rachel. Thank you so much for helping me with this research. And the REEU team, Dr. Jaime Pinheiro, Liz Garofalo, and John Galvan, and Caitlin Alshauskis, thank you so much. This was like a really cool opportunity in my scientific career. So this was just, yeah, very special for me. So I wanted to thank you all. And if I'll take any questions, if anyone has any questions, but if not, we can move on to the next person.
Moving forward, we have Jasper Richard. He is a environmental conservation major. He is from Mount Wachusett Community College. I am very proud of this individual. He is going to present his research next.
Hello, everybody. I'm Jasper Richard from the REEU program. Like John said, I'm currently a student at Mount Wachusett Community College, and I'm about to begin my second semester as an environmental conservation major. So this is my presentation titled Assessing Nematode Communities by Abundance and Diversity. Before I start, I want to give a huge thank you to John Galvan and Dr. Robert Wick. I wouldn't have been able to do this without them. So just a quick overview here. I'll start with a light introduction into nematodes, mostly go over the trophic groups. I'll go a little bit deeper into diversity and identification. I'll talk about the actual effects these nematodes have on soil systems, and I'll wrap it up with a little bit of methodology and just kind of my basic lab procedures. So first of all, what is a nematode? Nematode is a multicellular microorganism that occupies virtually all terrestrial areas as well as Arctic and aquatic areas. It's also the most abundant animal known to science. There are estimated to be around 57 billion nematodes for every human on Earth. And this is Nathan Cobb. He's known as the father of nematology. He was born in 1857 in Spencer, Massachusetts, and he's known as the father of nematology because he provided the foundations for nematode taxonomy and described over 1,000 nematode species before his death in 1932. And that quote at the bottom kind of gives you a decent idea of what we're looking at in nematology if you guys want to give that a read. So trophic groups. These are the trophic groups. Trophic groups just classify nematodes based on what they eat. So we have bacteriivores, fungivores, plant parasites, predators, and omnivores. And that Tylenchus in the middle is kind of more or less the most common nematode you'll see throughout soils. So what are the actual functions of these trophic groups? So bacteriivores will collect and eat bacteria with a complex mouth structure. Fungivores pierce and feed on fungal hyphae, extracting cellular fluids with its stylet, similar to plant parasites. Plant parasites use its stylet to puncture and feed on the interior of plant roots and tissues. Predators survive by consuming microorganisms and other nematodes, and omnivorous nematodes feast on a multitude of food sources, so they tend to get very big as they have many options. So how do they actually affect soil systems? They increase nutrient cycling, organic matter decomposition. They're great bioindicators of soil health, and they are essential to the soil food web. So nutrient cycling. Nematodes increase nutrient cycling by making nutrients like nitrogen and phosphorus more available for plant uptake. And since they often have a higher carbon to nitrogen ratio than what they eat, they excrete the excess. Organic matter decomposition. Nematodes break down soil bacteria into smaller pieces, making it easier for microbes to decompose organic material further into nutrients. Bioindicators of soil health. Nematodes are known to respond to changes in soil conditions like pollution, nutrient levels, and physical changes like soil structure, aeration, and water infiltration. Essential to the soil food web. Nematodes also occupy various trophic levels in the soil system, acting as prey and predators, which keeps the soil food web afloat. And effects on farming. Their activity can be used to assess the impact of management practices like tillage, pesticide application, and soil amendment. So my main research question for this project was, do nematode communities differ based on crop type and location? And if I was to study this further, I would look into the variation based on management practices such as pesticide application, soil amendment, or fertility programs. These are some of my basic procedures. So after I collected my 9 soil sample plugs from Cold Spring Orchard, from blueberries, grapes, and apples, I spent about a week in the nematology lab performing extraction. After that, it was about another 2 weeks in the nematology lab doing microscopy, just kind of identifying my nematodes based on trophic groups and their taxonomy. Then it was reorganizing all that new data into charts and graphs so I can see it, and then transferring all that new research to my final research paper in this PowerPoint presentation. So this is an aerial view map of Cold Spring Orchard. On the top is where I got my apple soil samples from block X1. And on the very opposite side of the orchard is block 39, where I got my blueberry samples. And on the right is where I got my grape soil samples from block G1A. This is kind of some of the basic equipment for cob sugar flotation and centrifugation method. We have soil samples measured into 25 cc portion, just makes it easier for processing. We have my 3 nested sieves along with a 1-liter beaker used for sifting and separation. And on the bottom left, we have a centrifuge, which separates the nematodes from the soil debris based on density. And on the bottom right, we have my labeled test tubes, which collect nematode samples after centrifugation. This is just kind of my data. It shows the number of nematodes that I counted per soil sample, categorized into their trophic groups. Just to kind of wrap it up, the first trend that I noticed in my data was that the number of nematodes in each sample increased the further away the sample was from the rootstock. This could be because of pesticide application or a soil amendment designed to keep microorganisms from causing damage to root systems. Additionally, the populations in each sample had significantly more plant parasites than any other trophic group. Thank you, guys.
Okay, moving on to the next person. We have Rahim Mai. He is a computer science major. He is a senior this year, or he's going to be a rising senior. And I'm really glad that we have somebody other than an agriculturalist doing the sciences that we do, because we need computer science, we need microbiome, we need economics. So everybody, please welcome Rahim.
All right, everybody. Hello. My name is Rahim Mien. I'm a computer science major. I'll be a senior in like 6 days, I think. So basically a senior already. But today I'll be talking about my research with broccoli and drone imagery. So why are drones used in agriculture in the first place? In order to think about that question, it's first important to understand the history of drones in agriculture. So it first began with the Yamaha R50. You know that company that makes those fancy pianos? Same company for some reason. They made a drone for agriculture. So it was used for pesticide application. So it was able to basically go through large rice fields and apply pesticides really quickly compared to manual application. And it also prevented the exposure of pesticides to the actual, people spraying them. So it was able to save on time, resources, and labor. And that was released back in 1987, and it's been like 40 years since then. So it's been quite a while. So drones have been getting some upgrades, and they're able to do a lot more than they previously were able to do. One of those things is they have cameras attached to them, and those cameras are able to take pictures of large fields in a really short amount of time. And with those pictures, with those maps, you're able to get a lot of data from them. And one of those data that you can get from them is something called the Normalized Difference Vegetation Index. Bit of a mouthful, but it's just NDVI. So what is NDVI? NDVI is basically a measure of the vegetation of an area. Effectively, it measures how green an area is. And it does so by using this camera, a special camera called a multispectral camera. And this camera, what it's able to do is we're able to see visible light. I can see green, black, blue, all those. But this camera can see more than that. It can see near infrared radiation, which we can't see. And chlorophyll, what gives the plants the green color, reflects that near infrared radiation. And so it measures that radiation, and the more near infrared light reflected, the more chlorophyll a plant has, and the more vegetation an area has. And that scale of NDVI is measured from -1 to 1, and a higher NDVI value indicates more vegetation. So here's an example of an NDVI map. You have the broccoli field on the left-hand side, and you have the NDVI map on the right-hand side. And you can see that the purple, it's basically where there's nothing. But the yellow, that cool part, that's where all the broccoli is. And so my question was, if NDVI measures greenness, How does that correlate with a plant like broccoli? Because broccoli is all green, right? You look at the broccoli stem, the leaves, the head, it's all green. So it's all going to contribute to that NDVI value. And how does it correlate between different stages of broccoli development? So those were my two questions that I want to research. And the way I did that was Dr. Jianyu Li here at UMass Extension had a existing broccoli field he was doing research in, and he allowed me to conduct my research there. And there was a drone that was flown every week on the Wednesday of each week, and it collected those maps that I showed previously. And I used 3 separate plots in the broccoli field, and each plot was a different cultivar. And every plot also had 30 plants. And so the way that I compared the broccoli head growth to the NDVI is I first measured the diameters of the broccoli heads. So unlike me, the broccoli head isn't perfect. The diameters, it's not a perfect circle. So I took 2 diameters. I took the longest diameter, and then I took a second diameter that was perpendicular and across from it. So it kind of made like a plus sign for all you math people, So I was able to get the surface area based on those 2 diameters. And then I was able to calculate the average head surface area of each plot. And then with that average head surface area, I compared that to the NDVI of the plot too. And so I did that for each cultivar over 4 weeks. And then once the drone got those maps of the broccoli field, a software called Pix4D Fields was used to generate the NDVI maps. That was provided by Dr. Lee, and it's not free, so thank you. And then QGIS, which is free, was used to basically analyze the maps. So Pix4D Fields made the maps, and then QGIS, which is a GIS software that analyzes maps, was used to get the NDVI. And my hypothesis was, as time progresses, the NDVI is going to increasingly correlate positively with the broccoli head growth. Because basically more time equals more growth equals bigger head equals bigger NDVI because more green. And was I right? No, I was wrong. So here's my data tables that basically insight of how I did it. I have the head surface area. I have the NDVI for each of the plots. But let's look at the graphs of them. So here's for the 2 cultivars, Eastern Crown and Castle Dome. You can see here there's a positive correlation for the first 2 weeks. So as the NDVI grew, so did the broccoli head surface area. However, at week 3, it started to show a negative correlation. So the NDVI would shrink, but the broccoli head size would grow. So it wasn't really that right. But one part I was right on, for these 2 cultivars, the NDVI meant a higher NDVI meant higher broccoli head growth. So at least I was right there. But then along came the cultivar Millennium, which made me 100% wrong. So this one had the highest NDVI across all the cultivars. However, it also had the lowest head growth amongst all the cultivars too. So yeah, I was kind of wrong. And then also, it kind of broke the trend too. So for the other 2 cultivars, the first 2 weeks showed a positive correlation. However, for this cultivar in week 3, the NDVI slightly grew by like around 0.1. So it was a positive correlation for the first 3 weeks, and then a negative correlation for the final 4th week. And the reason that may be so is because of the way that NDVI is calculated. I mentioned before that's calculated using near infrared light. And the more near infrared light, the higher the NDVI is. So for a plant like broccoli, chlorophyll isn't evenly distributed amongst all the parts. The broccoli head isn't going to have the same amount of chlorophyll as the broccoli leaves. In fact, the broccoli head has around 5 times less chlorophyll than the broccoli leaves. So if you're taking a picture from above and the drone is capturing the plant and the more of the plant is taken up by the broccoli head, then the broccoli head is going to contribute more to the NDVI value than the leaves. And if you have less chlorophyll in the head, then you're going to have a lesser amount of near infrared light reflected. So a bigger broccoli head could contribute to a lower NDVI value because you have less chlorophyll in the actual image you're using to generate NDVI. Another reason, a few couple reasons, is weeding, right? So it's not like a buffet. You can't pick and choose what you want to contribute to NDVI. It's going to be everything in the area. So weeds have chlorophyll. They're going to contribute to NDVI too. So if you weed more on 1 week or there's more weed growth on another week, that's going to change the NDVI week to week. Another thing is pests. So for an example, in this picture, right? You can see the broccoli on the bottom doesn't look that great. You know, there's a bunch of holes in the leaves. So pests are going to eat the leaves, which is going to eat the chlorophyll and reduce NDVI. That also connects to my point about how I use surface area to basically compare to NDVI, which may not be the best measurement for a week-to-week analysis because like with that broccoli, sure, it's going to have a big surface area, but it's also not really going to be that marketable. So a different measurement for a week-to-week analysis might have been better for future research. Another thing to keep in mind with NDVI is that you got to take it at the same time, right? The sun is going to be finicky. It's going to create shadows, and those shadows can cause differences in lighting and then cause differences in NDVI. So you have to be wary of that. But yeah, those were some of my considerations and limitations. All right, and for acknowledgements, thank you for Dr. Lee for providing me the research field and the software. It's like trying to make a pizza without, bread, sauce, cheese, or an oven. You can't really do it. So thank you. Seven Liu from Dr. Lee's lab also showed me how to generate the NDVI maps in Pix4D fields. And he also flew the drone and like drove me to the field like for most of the week. So thank you for that. All the members of REEU, John Galvan, Caitlin Alshauskis, Samina Mian, Dr. Liz Garofalo, and Dr. Jaime Primierot, thank you. And all my fellow interns, thank you.
Okay, moving forward, we have one more person. We're going to take a short break so you guys can ask questions. This next person is an actual agronomist, just like I am. So I'm very happy to be announcing this next person. This is Grace Brenny. Sustainable food and farming major. She is a junior. Please welcome on board. Grace.
Hey everybody, my name is Grace Brenny. I'm going to be a junior this year, and I am a sustainable food and farming major. So this summer, I got really interested in organic repellents and pesticides. So my research is titled Nasturtium and Garlic Extract as a Repellent Against Apple Maggot Fly. So let's take it back a little bit. Back in my young high school days, when my Future Farmers of America application was rejected for being too crumpled, I had no idea about agriculture. I really wasn't aware how organic systems work. So in my mind, I was like, they don't spray anything. They don't put anything on there. They just plant it, they leave it, and it's all beautiful and wonderful. And I was so wrong. I was so wrong. So both organic and conventional farming uses repellents. In fact, it is almost impossible to farm without some sort of repellent or deterrent. And even bug spray is actually classified as a pesticide. Anything that can prevent, destroy, or mitigate pests is classified as a pesticide. So that's kind of interesting. So these are some different types of organic repellents. You have physical kinds, you have essential oils, and you can also have pheromones, which can confuse bugs or affect their nervous systems. So the pest I wanted to focus on is apple maggot fly. I actually wanted to look at this weevil called plum curculio, but I missed the emergence time. So I chose apple maggot fly, and they actually started becoming one of my favorite bugs. So the way apple maggot flies life cycle work is they emerge in the summertime, they go and they find an apple and they lay an egg in it, and then the larva hatches in the apple and it eats the apple, which can be very detrimental to the crop. The larva turns into a pupa, the pupa drops out of the apple and overwinters in the soil to emerge again in the summertime to start the whole thing over again. So I wanted to see if I could find an essential oil that would repel this pest. So for the main organic repellent for this is kaolin clay. It's a physical barrier that is sprayed on the fruit, but it washes off every time it rains, so it can get kind of expensive. So I was like, maybe I could try a repellent, see if that works. So I chose nasturtium and garlic essential oil. And I did this because home gardeners often plant nasturtium around the perimeter to keep pests away. And they also plant garlic next to their cash crops to try and keep bugs off. So I was interested to see if this concept would translate over to essential oils. So my objective is, are these essential oils effective in repelling apple maggot fly? So onto my methods. So this is my experiment design. I sprayed apples, and these apples will then put in insect cages. I had a choice test, which had all three groups in the same area, and a no choice test. So this only had one apple to one cage. And so to start off my experiment, first I prepped my apples. When you go to the supermarket, they're all beautiful and shiny, and that's because they have wax on them. And some apples naturally produce wax, but didn't matter. I had to scrape it all off because the flies don't like it. And it can't be like washed away with a detergent, so you got to physically scrape the apples for hours and hours. And then I started making my essential oil concentrate. It's a mixture of water, oil, and emulsifier. So you have to make it the day of because it breaks down. And then, so those little containers are full of sugar water to keep my flies alive. And there's me spraying the garlic. It stunk really bad and I messed up the whole basement, but it eventually went away after a while. And then this is a great example of my choice cages, and these are my no choice cages. So I ran the experiment for about 44 hours, and then after the experiment was done, I would take the apples out. So there were 15 female flies in the choice cages, along with 6 male flies, and 5 females in the no choice cages with 2 males. And this was just to ensure that they were able to lay eggs for the wound counting. So I took the apples out, I incubated them in plastic containers with sand at the bottom so I could see when the pupa falls out. And then for hours, I looked under that big light first to see if I could find any anomalies, and then I looked under the microscope to count the wounds individually. So my hypothesis was that my nasturtium essential oil would be more effective than the garlic one and the control group. And was I right? No, dead wrong, dead wrong. The nasturtium actually was almost worse than my control group of distilled water, while the garlic was a very low wound count. And this is for my trial 1. Maybe I was right for trial 2. Wrong again. The nasturtium failed miserably. It was almost more attractive than the control group, while my garlic actually performed great and had very low wounds on the fruit. So this is my overall count of wounds on the fruit. As you can see, the nasturtium is a little bit more attractive than the control, and then the garlic is very, very low, having like 4 or 5 wounds overall. So this is my mortality of flies. So I had a lot more flies die in the second test for some reason, and they only died in the choice test, which was interesting. No one in the no choice cages, they were all fine. So I think this is because of the age of the flies. I kind of had a hard time finding a happy medium. So, but my results. So, an interesting thing to note is that the garlic residue I sprayed stayed on the apple for up to 4 weeks, and I could still smell it like even after 4 weeks, which is very interesting. And some future research could look into if water washes this off, like it could also look into if the flies are repelled by the smell or the residue. So I thought that was interesting. And another thing to note, does this garlic spray affect the apple's taste? I did not even consider this, so that could be kind of bad. But that would be good to look into. Some limitations I faced, the 44-hour trials, it was up in the air because the building's locked and you got to get to the flies at a certain time, but it was around 44 hours. And then again, the age of the flies. My first group was maybe a little too young. My second group was maybe a little too old. So I just kind of had a hard time finding that happy medium, but I think it worked out in the end. And yeah, why is this important for agriculture? When farmers have more options, you have more options. When farmers are able to have a full toolbox, you can have a better, more sustainable, supported food system. And yeah, and this is us at the farms. And just thank you all. Thank you, everybody who helped me along the way. Thank you, Dr. Pinheiro. Thank you, Liz Garofalo. Thank you, John Galvan. Thank you, Ajay, for helping me with my concentrate. And thank you, Suhong, for helping me take care of the flies. Thank you so much, Caitlin and Sabina. You guys gave me so much support. Thank you to my awesome REEU interns. It was so fun. It was my first summer away from home, and you guys made it really awesome. Thank you to my awesome roommates and my family for coming out, my boyfriend too. So yeah, thank you all so much.
All right, everyone, we're going to be moving on to the second half of our symposium. Our next individual here is Jacob Zeller. He is a microbiology major. He is a senior this year, right? No, junior? 2027. That makes sense, right? Horrible math. I worked personally with this individual. I'm very, very proud of him. He has great lab skills and a bright future. Jacob Zeller.
Hello, everyone. Hope you enjoyed the break. Thank you to John for that good introduction. Yep, my name is Jacob Zeller. I am a microbiome major, and I'm working towards my bachelor's, about to be a junior. So, as a microbiology student, I knew that this summer I wanted to work with bacteria on my project. That leads me to my project title, The Effects of Biochar on Remediation Capabilities of Isolated Bacteria from Glyphosate-Treated Soil at the University of Massachusetts Amherst campus. I'd also like to give a thank you to John Galvan and also Dr. Robert Wick for helping me a lot during this summer, really working through my ideas and giving me a good direction for where I want to take my research. So, what is a problem I am trying to solve? As a microbiologist, I'm really concerned with the environment, and also I'm looking to hopefully create some sustainable solutions to problems that might have some solutions that are a little more damaging to our environment. Specifically, I was working with glyphosate this summer, which is a broad spectrum herbicide. Many of you might know it's a common ingredient in Roundup spray that's used in both agriculture and also residential settings. You can see here on this photo, there's this strip of dirt and rocks. That is in an orchard where herbicides like glyphosate are sprayed, and they're used to treat a bunch of the small little weeds around a rootstock of a tree, weeds that might impact the amount of nutrients that can reach your tree that you want to have fruit. So glyphosate is very great at treating these weeds, but because it's also non-selective, it can affect other plants. In this case, it can also affect your fruiting trees, which is really bad if you're trying to get the most growth that you want out of your fruiting trees. In this case specifically, there was a study done on Jonah Mac apple trees that suffered a winter injury after glyphosate was sprayed a season ago. So you can see these brown leaves at the bottom of the tree. They're died off. They are not producing any energy for the plant, and it's affecting the photosynthetic rate because they're not creating any energy and they're all dead. So it's going to affect, your fruit yield. This is not great, of course, and there are some solutions that have been developed to remediate some soil and try and clean it up across seasons so that you avoid some of these sublethal injuries, many of which are chemical and also mechanical treatments. These can have some issues, though, with efficacy and also cost, so they can be pretty expensive to try and clean up your soil. But I really wanted to work on a sustainable solution to this kind of problem, so I turned to bacteria. Bioremediation is a method to remediate a pollutant in soil or in water, where you use a living organism to break down the pollutant in your environment. It'll chew up the pollutant, break it down. It'll use the elemental carbon or phosphorus in your pollutant as a food source. So you can use bacteria, fungi, and algae to break down pollutants. In my case, specifically, glyphosate is a great source of both carbon and phosphorus, and will hopefully be able to be a source for bacteria. And if you'd like to treat your soil by using a bacterial remediation technique, you would need a lot of bacterial spray because the bacteria have to be in direct contact with this carbon in order to break it down, metabolize it, and use it as a food source to replicate. But this can be pretty expensive because you need a lot of spraying and you need to cover each place that has been treated with your glyphosate. But this is where adsorbents come in. Adsorbent, in this case, biochar, is a method to adsorb pollutants into a concentrated area. Biochar is used, it's pyrolyzed woody material such as trees or rice husk. It's got a lot of pores in it, and it's got these matrices that are able to trap pollutants inside of the biochar particles. So anything that's a pollutant in water that flows through the biochar will be trapped onto the matrices and into the pores and be concentrated in a small area because you don't need a lot of biochar to do a lot of adsorbing. But coincidentally, biochar is also a very great habitat for bacteria. It's got a lot of these pores, as I described, so water can flow through with nutrients and waste can be washed away as well that the bacteria might produce. So separately, both adsorbents such as biochar and bacteria have been studied very well to have high rates of cleanup and also adsorbing properties, but little has been done to study the combination of both biochar and bacteria together when trying to remediate a field. That's where I thought I could do this project over the summer, and I did a small in vitro study so that I could find a way to either improve the remediation process or make it a little cheaper. This is where I started with a soil isolation. I isolated soil from the UMass campus because it is local. If you're trying to find a bacterium from soil and you want to use it on a farm setting, you don't want to introduce a strain or a colony that's really far away because it might have some ecological impacts because it's not native to that area. So I started with the UMass campus. Specifically, I talked to landscapers that use glyphosate on flower beds around the UMass campus to make them look pretty. They clean up some of these small weeds that you see around here. and they're commonly treated with the glyphosate over the course of the summer. So I thought I'd take soil from these areas because such a common spraying location of glyphosate means that the bacteria that grow around here should hopefully be able to break down the glyphosate in the soil. It's a very commonly sprayed source of carbon, so it'd be very beneficial to the bacteria to be able to break it down in the soil and grow with it. So I took this soil to a depth of 6 to 8 inches from UMass. I plated it. I diluted it out and I plated it so I could find some isolated colonies that I could hopefully study further. Here you can see on the left, there's the soil samples that have been diluted out. The much darker ones are like pure soil. I diluted them out so that I could plate them on this non-selective media, King's B, and I could take these colonies, I could swab them and use them for later usage so I could pick out which colonies I think look unique and might have a great chance of degrading my glyphosate from the soil. So with these isolated colonies, I went ahead, plated them on a minimal salts media or MSM. Minimal salts media has no carbon source of its own. It's just nutrients that the bacteria will need to grow, but I introduced my own carbon source in the form of this glyphosate here. You can see this molecule is in a organic chemistry formation. All of these bends that don't have a letter are the carbon. So carbon's essential for everything to grow, and I'm introducing this glyphosate in order to allow my bacteria to have a chance to grow. As a selective media, basically anything that cannot break down the glyphosate will not be able to survive because it's the only source of carbon that I introduce. Since everything needs carbon, if they can't break it down, they won't survive, and anything that does grow, I know can break down the glyphosate. This is my selection, and I was able to see that after plating 48 different colonies, only one was able to survive on my glyphosate, which is great. Only needed one colony to move forward with my project, and I knew since it could grow that I could use it later and on more glyphosate-related experiments. So I took this colony, I streaked it back onto King's B for preservation, and I did a quick gram stain to see it was a gram-positive cocci, which is just a simple morphological analysis. Here it is under the microscope at 1000x. You can see these dark purple dots around here. That means that they are cocci because they're a little circular, and the purple means that they are gram-positive. So from here, I could actually begin my experiment with my treatments. This is very exciting. I was able to move forward and do these experiments in flasks. So I had 3 unique treatments plus a control, and each of the 3 treatments had 3 replications. These are all done in 125 Erlenmeyer flasks with 100 milliliters of media. There was first my control, second was just my bacteria treatment on glyphosate. The third was just bacteria alone with the glyphosate, and the fourth was a combination of both my bacteria and biochar. The whole point of my experiment here and my hypothesis is that when I combine both my bacteria and biochar together in the flask, they'll have a much faster growth rate, which also means a much higher rate of degradation of glyphosate. Because if they're growing fast, they're breaking down my media fast and hopefully producing less toxic end products. In this case, they were all incubated on a shaker incubator at 28C for 150 RPM. These are just some of the materials I used. I had Roundup, Falcon tubes to grow them, media, biochar, a soil plug, some filters, and I ended up collecting data with spectrophotometer and cuvettes. So here's a quick graphic of my experiment. On the left, you can see the control just had Roundup and my minimal salt media with nothing else in it. The second was just the biochar. The third was just the bacteria alone. And the fourth was everything, but with both bacteria and biochar. My hopeful experiment, they would have great growth. So here it is on the shaker incubator inside of the incubation unit. On the far left, it's just the biochar, then the bacteria alone, the combination, and my control. You can see both the biochar and the combination flask were very dark because these biochar particles are floating around, hopefully absorbing my glyphosate inside my media. So to collect data, I decided to use a spectrophotometer, which is pretty preliminary, but it's great at analyzing the rate of growth of a bacterial strain. So I took a 1 milliliter aliquots from my experiment over the course of 3 days so I could analyze the growth rate over time as time progressed. I would take data at 0 hours, 6, 24, 30, 48, 54, and 72 hours. These are pretty irregular time intervals, but I'll describe them later. I took these aliquots and I centrifuged them at 3000 RPM for 2 minutes in order to pellet the biochar at the bottom of my sample so I could take just the liquid and hopeful bacteria. They were all analyzed in the spectrophotometer at 616 nanometers. Now, if you're not sure what a spectrophotometer does, it basically measures optical density. Optical density is a value from 0 to 1 that describes how much light can penetrate through a sample. The spectrophotometer shoots a light through your sample and it has a sensor on the other side. And depending on how cloudy the media is, you'll be able to tell basically how much light can get through the media. So when bacteria multiply, they become there's a lot more biomass in the flask, and it becomes a little bit more cloudy. So a higher reading means that the bacteria have grown a lot more. And I was able to take this data and use it as a proxy to determine basically the rate of glyphosate degradation. Essentially, what this means is if the bacteria are able to multiply, replicate very quickly, they could break down my glyphosate very quickly, and they are therefore much more efficient at degrading glyphosate. Whereas a slow rate where they don't become very cloudy quickly means that they're having a harder time trying to break down my media. So here is my data. Pretty confusing, right? Well, let me break it down for you, much like the glyphosate in my experiment. Thank you. So here's my treatment alone with just biochar. It might look great that it has a positive slope, but in actuality, this is an abiotic experiment. So there's no bacteria in here, which means theoretically, there should be no slope at all. There should be no change in time of adsorbance because nothing's growing, so nothing should be becoming more cloudy. But this is the biochar alone. It stands pretty low, around 0.05 to 0.2. This already highlights some of the issues with taking spectrophotomic data when you have a cloudy media, such as biochar, because it can affect some readings. So this is something to take into account before I go on. But this is my combination of both biochar and bacteria. Looks great. At about 48 hours, you can see that there's a big spike in growth, which is amazing. So in the beginning, though, there's a bit of a plateau as the bacteria are becoming acclimated to their media. But by about 48 hours, they have a much higher growth rate and a very great positive slope. This is great fast growth, but however, my bacterial strain alone had a very, very hard time acclimating to this media. If you're not a microbiologist, you might not know, but when you introduce a bacteria to a new media, they take a little while called the lag phase. It takes them some time to get used to their media before they can begin breaking it down and replicating and increasing that adsorbance value. So here we can see only at about 72 hours do they really begin to take off, and even so, still very slow. But this basically means that they had a much harder time adjusting to their media, and it took them a while to begin breaking down the glyphosate because they weren't very comfortable with the media. Whereas you can see again, at about only 48 hours do the combination experiment really become comfortable, and they're able to take off really quickly into much more exponential growth. So what does this all mean? Well, I took a trend line in Excel and I assumed that each plot was linear. I plotted a trend line and I took the slope and once again plotted it into this bar chart. So these are the slopes of the growth. So this is basically a measure of the growth rate over my 72 hours. You can see in the control, they're at 0, so that's great. Nothing grew in the control. But of course, in the biochar, as I said, there's a little bit of growth, which is not really ideal because theoretically, they should also have a similar 0 slope. But regardless, you can see with my T1, 2, and 3, that is my isolate. It has a very, very small slope, so it had a really hard time growing, and you can tell from the other graphs that they're pretty flat. However, my combination of BT1, 2, and 3 had the highest slope out of all my treatments, which is great because this theoretically would prove my hypothesis that the biochar is providing a good environment and allowing that bacteria to really grow, replicate, and break down my glyphosate pretty quickly. But there are plenty of limitations and a couple of sources of error in my study, starting with the biochar separation. As I described, that positive slope on the biochar is not ideal. It kind of highlights that taking these aliquots when you're trying to measure the optical density, the coloration of your media, when you have this dark biochar, it can really provide some inconsistencies in my data. So it was interesting to note that perhaps spectrophotomic data might not be the best, but it's a good preliminary to give a little bit of an idea of how this experiment might run when you take some more chemical data. In addition, my schedule sampling was a little bit inconsistent because the Page lab was only unlocked for so many hours during the day. I unfortunately could not come in at 12 a.m. to take data, but I took one regardless in the morning and the afternoon. In addition, the 72-hour time span was a little bit limiting because, as I described, the bacteria, they only began to take off at the very last data point. So it might have been a little bit better to see a little more growth as time went on, and I could have seen them reach that exponential growth phase. In addition, there was also only 3 replicates of each, so it kind of takes away from some of the statistical significance, but more experiments could be done in the future to kind of give a little bit more foundation to my data. And lastly, as I described, the positive slope in the biochar samples kind of gives you an idea on why maybe spectrophotomic data is not perfect for this kind of experiment. But for the future, I think I'd like to look at maybe some more chemical methods of analyzing my data. Specifically, GCMS would be great, mass spectrophotometry. Basically, I would take a sample before I introduce any of my treatments and also after, and I'd be able to see the actual changes in that glyphosate media, basically how much is actually truly broken down when you have bacteria versus biochar versus a combination of the both. That would be great to explore in the future and more of a more in-depth experiment. And also sequencing on my bacterial strain would be great. I did a gram stain, which provided some basic idea, but doing this sequencing would give me a better idea on the genus and also the species of the bacteria that I isolated and give a more, basically just a lot more information on this bacteria and how it might survive in an actual farm setting. So thank you so much to everyone. I'd like to once again thank John Galvan and also Dr. Robert Rick for really helping me out over the summer, and also the rest of my REU team. You guys were great, and thank you so much.
I am happy to announce these next 2 individuals. Delia Ekblom from Greensfield High School. She is one of 2 of our high school students that joined this program this year because our program is expanding, and she has some great research. Please come forward. Delia Ekblom.
Good morning, everyone. I'm Delia Ekblom. Like John said, I am one of the first high schoolers to join this program. So this summer, my study focused on the effects of microplastics on the growth of cucumber plants. And before I go into this, I want to say that Caitlin Alashowskis was so helpful for this entire process. She was with me in the greenhouse most of the time I was there. It was so hot. So she's amazing. So what are microplastics and why do they matter? Microplastics are plastics that have broken down to a size of about 100 nanometers to 5 millimeters. They can pollute the environment in the air, the soil, and the water. And they also have the potential to negatively affect plant growth, plant yield, and the health of human beings and animals. A couple of the key points that I took away from the literature I reviewed this summer fall into 3 categories: potential health impacts, environmental influences, and effects on growth. So for potential health impacts, the research around this can be a little iffy because there's not a lot of it done, and some of it that is done on animals. But so far, some of the things that have come about are different types of cancers, which are very occupational, so it depends on where you're working, decreased fertility, DNA damage, and gut barrier dysfunction. Some of the environmental influences are changes in soil bulk density, and it can cause heavy metal contamination in water. And some of the effects on growth, it can inhibit growth by physically blocking roots. It can disturb photosynthesis by messing with the chlorophyll A and B ratios, and it can reduce yield of many different types of plants. So my research question this summer was, how do microplastics typically used in farmlands affect the growth of cucumber plants? And I had 2 hypotheses. One was that cucumber plants affected by one type of microplastic would exhibit reduced growth compared to the control group with no microplastics. My second was that cucumber plants exposed to 3 types of microplastics would experience much greater reduction in growth than those exposed to one type. I had 3 goals going into this research. One was to conduct further research so that people might push for more sustainable alternatives to plastics. The second was to demonstrate the potential risks of using plastics in farm systems. And the third was to give farmers and others interested in growing their own food enough knowledge to make safe decisions for themselves and for others. I grew cucumber plants for about 2 weeks in just like a regular cell tray, which was plastic. And then after about 2 weeks of growth, I transplanted them into pots like those, and I divided them into 5 groups of 5. One group was my control with no microplastics. One group had polyethylene, one had polypropylene, one had polyvinyl chloride, and one had all 3 types of microplastics. So I chose these plastics firstly because they were what I had to work with, but it ended up being very convenient for me because these plastics are all typically found on farms. So polyethylene, I believe, is used, yes, it's used in anything flexible. So irrigation tubes, drip tape, stuff like that. PVC, PVC pipes. And then polypropylene is used in plastic woven bags. So I took measurements twice a week, and I took measurements of the height of the plant, as well as the length and width of the newest leaf on the main shoot. And by length of the leaf, I mean from the petiole to the tip of the leaf. So this is my first graph. This is the average stem height over my 6-week period. It says 12 because it was twice a week, so 12 days. For most of the growth period, my group that had all 3 types of microplastics was my tallest group. And then my lowest group was usually my control group. So what this says to me is not that my group with all 3 microplastics did better, but that it did worse because when a plant has less nutrients, the cells stretch. So rather than being condensed and stronger, it's longer and weaker. And then my next 2 graphs are the measurements of my leaves. This is the base, so from the length. This one didn't really have any significant trends, and neither did this one. So in the future, if I or somebody else were to do another research project like this, I wouldn't include measurements of leaves. So there were some visual observations that I saw that could be relevant to microplastic contamination. So this is a picture on my right is polypropylene, and the left is my control group. And you can see that the polypropylene group has flowers on it, and the control group doesn't. So my polypropylene group flowered maybe a week or so before my control group. And from then, the rest of my groups with microplastics flowered before my control group. And then also the my leaves are yellow, basically. So you can see it's a little white on that side. It's pale around that little hole in the leaf. It's generally very pale. And if you look at my control group there, it's obviously much paler than those leaves. So what this tells me is that the microplastic exposure could have reduced the chlorophyll in the plants, which is something I have read. Some of the limitations I ran into this summer were time constraint. I maybe started growing my plants around the 4th or 5th week, so that was a bit of an issue, and also lack of research experience. But honestly, I feel like that wasn't that big of an issue because that's sort of the purpose of this internship, to give people who've never done research before an opportunity to try that and fail or not fail. And then also, the greenhouses had some temperature regulation issues. So it might have been 100 degrees or more during the heat waves this summer. So as you can see in that picture, that leaf is browning on that side. And then also I had some dying on the tips of some of my plants. So microplastics, regardless of this research, have been shown to have negative effects on plant growth, on human health, and on animal health. However, the data in this project did not really show that. So in the future, if somebody were to do research like this, I would recommend having more time and using different types of measurements to analyze the growth. So that's my references. And I also want to thank Caitlin again, but also everybody who helped this summer in REEU, the cohort, my mom, Elizabeth Garofalo, Jaime Pinheiro, and John Galvan. So thank you.
Now moving forward to her sister, who is also a Greenfield High School junior this year. Good luck in high school. Isabella Ekblom.
Hi, everybody. I'm Isabella Ekblom, and I'm a junior at Greenfield High School. This summer, the main question of my research project was whether or not Japanese beetles survived better on basil, beans, strawberry, or a mix of all three. So for a little bit of background, Japanese beetles originate from Japan, and they were found in New Jersey in 1916 in a plant nursery. Since then, they spread throughout the United States, Canada, and parts of Europe like Italy. They're a polyphagous pest, which means that they feed on a variety of host plants, and Japanese beetles specifically feed on over 300 different hosts. They can cause a lot of damage to cash crops, and that's a big problem for farmers. So the main goal of my research was to find which of the three plants in my project were the most attractive. And I also wanted to see whether the Japanese beetles survived better on a single host diet or a mixed host diet. My hypothesis was that the basil would be the most attractive because it had the strongest smell, and none of the plants were flowering, which is why I believe that. And I also believed that on a mixed host diet, the Japanese beetles would live for longer. So the materials that I used for this experiment were 12 insect rearing tents and around 225 Japanese beetles, which I collected from the permaculture gardens at UMass Amherst. And then I also needed 6 basil plants, 6 strawberry plants, 6 bean plants, and 3 small water cups for my control. Here you can see my choice test, which had a basil plant, a bean plant, and a strawberry plant all in one cage. And then here you can see my choice tests, which were my non-choice tests, excuse me, which were my basil in its own cage, my bean in its own cage, and my strawberry in its own cage, and also a control of only water. So the data that I collected, I collected data 3 times a week, being Mondays, Wednesdays, and Fridays. And the data that I collected was the number of dead beetles, the number of beetles on the leaves, and the injury. So for the injury, that was a scale of 1 through 4, 1 being no damage, 2 being little damage, which was 1 through 10% of skeletonization on the leaves, 3 being moderate damage, which was 11 to 20% skeletonization, and 4 being severe damage, which is anything above 20%. And that picture there just shows what I mean by damage and skeletonization. So this is my first graph, which shows the average beetle feeding injury on the bean, the basil, the strawberry, and the choice versus non-choice tests. This is you can see my choice test graphed, and the basil had a pretty high damage, and the strawberry had consistent damage throughout all 3 of my replications. As you can see, the bean had pretty low damage, pretty consistently. And then in my non-choice tests, you can see that my strawberry had the highest percentage of damage. And my basil also had some pretty high damage, but my bean again had the least. So this graph shows my beetle mortality rate over time. And it starts at 100% of the beetles being alive and goes down. So the control, of course, were the ones who died out the quickest. And the bean also followed that pretty quickly. And surprisingly, the mixed diet did not live for as long as I thought they would. And instead, the strawberry lived the longest and had the lowest mortality rate. So previous studies have shown that generalist herbivores survive better on a mixed host diet, which is why I expected a similar thing to happen with this experiment. However, the strawberry consistently proved to sustain the Japanese beetle's life for longer. Some limitations of my project were the quality of plants. At one point while they were growing in the greenhouse, they got spider mites, and so they all had to be replaced. And then also after the Japanese beetles started feeding on the basil, they sort of wilted and died faster than the other plants. And also I think in a future experience, I would start the project a little bit sooner because then some things could fall into place a little bit better. So in conclusion, this project aimed to find which of the 3 plants were the most attractive, and also to assess whether Japanese beetles lived longer on a mixed host or a single host diet. We found that strawberry was the most attractive, and we also found that strawberry was the most beneficial to the Japanese beetle's life. In the future, I would say a study involving a wider variety of plants and maybe more replications could be a little bit could provide more insight. And I'd like to thank Kaylin Alashowskis, John Galvan, Dr. Jaime Pinheiro, and Elizabeth Garofalo for being really helpful throughout all this process, and also the other REEU interns for just being super helpful. So thank you.
All right, guys, we have one last person, and this person is very, very important because they are not in agriculture whatsoever, but they are very important to what we do in our systems. This next person is Sophia Iftikhar. She is a resource economic major. She is a junior this year, right? She specializes in economics.
Thank you, John. All right, everybody, bear with me. I'm kind of under the weather, but I'll get through this quickly. All right, so my research was interested in exploring farmers' willingness to participate in codling moth mating disruption cost share programs. I will break all of this down. I also learned about these terms in a relatively short time span. So, step back, the overarching issue is with pesticides. I'm sure all of you know, are well versed in the damages that pesticides cause from an ecosystem level and on a social level. What I've learned through the course of this research is that there are ecosystem damages caused by pesticide application, whether physical or chemical. This extends to farmers who have to deal with them directly. And in the context of my research specifically, pests have adapted over time. Specifically, my pest of interest, codling moth, has developed to chemical applications, leading to higher expenses and greater health concerns towards farmers. This has led to the development of a technique called integrated pest management. It is to do with applying pesticides at a viable area and using alternative means of pest control as an economically viable way of dealing with pest damage. So this could be anything from cover cropping, trap cropping. I'm specifically interested in this concept of mating disruption. A really key component underlying integrated pest management is this concept of economic pest thresholds, which entails spraying when the damage of the crop exceeds the costs of controlling the pest. And what I mean by mating disruption is it is an integrated pest management technique. It is facilitated through installing pheromone disruptors on different blocks and orchards. This is basically by spraying sex pheromones that distract male pests from finding females and mating. And a good example of how this works is through the example of trying to locate a very specific kind of advertisement and then getting bombarded by Times Square. And so that's how it works in mating disruption. So my research is specifically focused on this mating disruption cost share program facilitated by NRCS, the National Resource Conservation Society by New Hampshire. They're looking at funding a sort of program in which they outline what mating disruption is. This specifically towards tree pests for apples, codling moth, and peach borer specifically, but I'm focusing on codling moth. And my interest is the application of a similar program in Massachusetts. So I should probably clear up what agricultural cost share is. It is when a government agency or third party pays for a portion of the implementation of a conservation practice. In this case, it is mating disruption. This can be through upfront payment of cost share funds. So say that they pay about 90%, they would be paying they're like, I'm going to give you $90, you can only pay 10. In addition to this, there's non-monetary provisions. They typically provide equipment and training as well to implement a program. And thirdly, again, it doesn't have to be 50-50. There's different cost share levels, ranging from 50%, which is half and half, to 90%. So my factor of interest is apple markets. So if you go to an apple orchard, there isn't one way you can have end use for the apples. There are supermarket wholesale apples, pick your own apples, and processing, which is cider apples. Supermarket wholesale, as my fellow intern Grace stated, are very shiny and pristine. You know, when you go to a supermarket, you don't typically pick ugly apples. It's very difficult to encounter a supermarket apple with any pest damage on it whatsoever. So the aesthetics are really held in high regard for supermarket apples. I think a step below that, I would consider that to be pick your own apples, which is where people can go to the orchards directly and pick them straight from the trees. There is no wax. Obviously, they probably choose ones that look pretty good, but there is no high aesthetic value to that to the extent of supermarket wholesale. And then thirdly, processing. It's interesting. So when you're creating things like hard cider and soft cider, alcoholic, non-alcoholic, usually pest damage, given it doesn't go to the extent of leading to premature rot, is allowed in processing cider because, especially in hard cider during the fermentation process, it gets sterilized. So aesthetics are not a major component because it's getting crushed up essentially. So drawing it into my hypothesis, it is holding other factors constant. The primary market the farmer operates in does not affect
cost share program participation. Now, there's many factors that impact farmers' willingness. Previous research indicates that farmers' knowledge of what the conservatory practice is a huge factor, as well as, ability to pay, their financial security. But I was particularly interested in seeing that there were different cost inputs in the different markets, how that would play into their willingness to participate, because there is differences in the price application of these markets. So for my data collection and analysis, I started with these targeted interviews with apple farmers, trying to gauge what factors influence their participation, what do they know about these things? Have they previously used mating disruption on their farms? And this was meant to kind of guide my later surveys that I would distribute to the growers in the region. This was very helpful. So I started off with a preliminary survey distributed at the Massachusetts Fruit Growers Association, MFGA. This resulted in about 27 responses. After being filtered out, I had 40 responses from there. And then later, I decided to develop a secondary survey because I found that I had made certain errors in my preliminary survey. However, what we'll go to later, I decided to count for them both in my later research. And this one was distributed via agricultural newsletter by Miss Liz Garofalo. And then later, to analyze my data, I did a Friedman-Wilcoxon test, which I will break down later. So here's an overview of the respondent profiles for my first survey. It showed that for the large part, direct-to-consumer, which is the market I chose, had the highest number of respondents. I think that's because it's a very broad market. It can encompass retail, it can encompass pick your own. So it's pretty broad. I mean, following it up, there was a pretty even split between wholesale growers and processing growers, and then very few did other, and that typically encompassed things like donation. The percentage of farm income from the main market was, for both surveys, actually the majority. This was around 77.39% and a little bit less in the second survey. As for the secondary survey, again, the direct-to-consumer-ish, I decided to, because it was an improvement on the preliminary survey, I decided to specify that broad categorization into pick your own, but similar responses still held. Pick your own farmers dominated the survey, even though the population was very small. The N was 10. And this, again, was another category I think that was very important in my results. It's the orchard size distribution for both the paper survey and the online survey. As you can see from these results, most of these orchards were large. There were never any responses that were under 5 acres or even in the 6 to 10 range. So these were largely major farms. So indicates some economies of scale and financial stability. So this is a broad outlook on the initial responses from my first paper survey. As you can see, there's a high likelihood of participation regardless of the markets they operate in. So this is taking into account all respondents. So at different cost share levels, we can see at the 90% level, people are very likely to engage in a certain program like this. And this continues even with the reductions in cost share coverage, such as at the 75% level, even though people were less likely to indicate very high likelihood, it was still a likelihood to participate in general. And this was also similarly seen in the second survey, where very likeliness to participate was a component in both the 90% and the 75% level. So now going into my statistical analysis, I did a Freeman-Wilcoxon experiment. This was to indicate statistical significance between the primary main market occupied by farmers and their willingness to engage in the cost share program. So I did this for each of the levels. I first did a Friedman to gauge whether there was statistical significance, to see if there was a pattern. And what I did find is that the p-values were strong enough to indicate that there were differences in how respondents chose based on their primary market. And then this is the results of the Wilcoxon experiment, and it did showcase that on average, the median scores for wholesale growers were slightly less than the other markets. And interestingly enough, this wasn't seen in the follow-up with the secondary survey. And this has led me to believe that beyond the other limitations of the survey, the broadness of the initial categorization of the markets definitely played into the ranking. So future study would definitely probably look at end use rather than markets as one of the factors. But it is still interesting to see that these differences were considered statistically significant for the non-parametric test. Again, this was a little bit more context. These tests were non-parametric because the sample size was that small, so I had to account for these. Could not do a correlation, obviously, for those factors because with such a small population, it could not be indicative of a simple correlation. And again, my question that I was trying to answer was, do different market types differ in their willingness to participate at a given cost share level? So going into my other limitations and considerations, again, sample size, 27 were reusable for the first survey with 10 for the second. There was an opt-in question format. Again, I did count incomplete responses given they had selected different cost share levels because, it was a long survey that we handed out towards the end of the day. So I guess nobody wants to answer a 3-page survey, which is good to note for future research. There was also non-random sampling. This was targeted. I mean, this research was facilitated by UMass Extension, so a lot of these, so who I was interviewing had a connection to UMass Extension and probably knew a lot about IPM in general, so there was generally a positive viewpoint of sustainable techniques. I guess future research would go outside of this scope to get more variable data because in my write-up, I did discuss that extension probably played a large role in the positive outlook towards engaging in a cost share program of this nature. And again, potential bias and purposive targeting, because it is extension adjacent. And of course, it fails to account for all factors. We couldn't control that. And then I just listed a bunch of, biases that are indicative of doing a survey. In the future, like I discussed, focusing on end use rather than market would be, I guess, more specific. I would break it down looking at cider apple specifically, and then maybe wholesale, keeping those things different. And yeah, I mean, but it is interesting to see that there was a significant showcase. And in the future, I would also find a way to actually collect discrete variables on farmer output and financial stability. Finally, I have some acknowledgements. Huge shout out to Alan Bill Rose of Red Apple Farm, Russell Brain of Park Hill Orchard, Tom Bashista of Bashista Orchards for doing the interviews of this part of the section. It was very important in informing the construction of my surveys, as well as the Massachusetts Fruit Growers Association for those who took the time to fill out my survey, as well as UMass Extension, the Stockbridge School of Agriculture's, obviously the REEU directors, John Galvan, Dr. Jaime Pinero, especially for staying with me late to knock out all the statistics, and because I had a lot of roadblocks in my research, especially when the amount of respondents were less than I had seen, was anticipating, as well as my fellow REEU cohort. Thank you so much for getting me through this program, and thank you all for coming.